Hello everyone, I'm Koyan and today we are going to learn how to answer question related to Form 4 Chapter 6 Cell Division. So basically we got learn about mitosis and meiosis and the application of mitosis and about cancer, tumor. So by looking at this first question, they show you 4 stages of mitosis. Then they want you to take the appearance of chromosomes seen in stage P. So let's see at the stage P, they got a chromosome, okay, and then basically you can see the is in form of chromosome, it's not in form of chromatin. So chromatin basically excluded. What about homologous chromosome? So homologous chromosome actually exists in meiosis only. Homologous chromosome is a pairing up of 2 chromosomes together, and it can be only seen in the meiosis situation. This is just mitosis, so all of them are just single. Yeah. And for sister chromatid, there are sister chromatid right here. This chromosome, it consists of two sister chromatid. So they have sister chromatid. And they want you to arrange the correct sequence of the stages in mitosis. So mitosis consists of P, prophase. So which one is actually prophase? Prophase is the part where the chromosomes are condensed, and this one is a prophase. What about metaphase? So metaphase means means they are arranged in the middle. So this is the metaphase where they line up at the equator. Followed by it's time for them to get separated. So when they get separated, say bye-bye to each other, they are pulled away. So away means anaphase. And last but not least is the telophase. So the last one is telophase right here, where the you can see that they start to form two individual cells. So the correct sequence is P, S, R, Q. So state the difference in the chromosomal behavior in stage R and S. So the importance of the mitosis, you have to at least know what are the chromosomal behavior. I know there are actually a lot of things to remember, like about what happened to the nucleus, what happened to the nucleolus, the centriole, what happened to sister chromatid, the centromere, the spindle fiber. Yeah, there are a lot of things. But if you, the most fundamental part you have to know is actually the chromosomal behavior. So I've actually written it out. The chromosomal behavior for prophase, metaphase, anaphase, and telophase of the mitosis. This mitosis. So prophase is chromatin. It will condense into chromosome. And then metaphase, they will align at the equatorial plane. Anaphase is where the centromere will divide. So the sister chromatid will separate and move to opposite pole. And last but not least, telophase, the chromosome will decondense back to a chromatin form. So stage R and S. What is stage R and S right here? Stage R is the anaphase. S is the metaphase. So what are the difference between them? So anaphase means they are away from each other. Metaphase means they are lining up in the middle, right? So at stage R, sister chromatid, they separate, and they are being put by the spindle fiber, put to opposite poles, while for us is the metaphase where they are lining up. So chromosomes, this one is chromosomes align on the equatorial plane. So the keywords right here is sister chromatids separate and align on the equatorial plane. Moving on to the next one is how this lizard, okay, able to escape from predator by losing its tail, right? And then they want you to explain how Patty can actually grow again after losing the tail, because they want to scare the predator, like why the tail suddenly can move, and then yeah. So this actually uses the application of mitosis. So the first thing you can exactly write, I mean you can directly write, is wire process of mitosis. Of mitosis. So mitosis is actually basically keep producing new cells, right? So it will produce sing will produce new cell. And this one is actually involving regeneration. That's why there will be a new tail that grow again. This is what the lizard have. I mean, they have this regeneration ability. But for us, yeah, when our hand patah, then yeah, already bye bye already. So we don't have this ability to regenerate a new hand out. So yeah, but basically the mitosis will keep, dividing to produce new cell. That's why they get to regenerate a new tail out. And moving on to the next question, it shows the uneven distribution of chromosomes during meiosis in process of spermatogenesis. So the sperm produced, which is the male gamete, have different number of chromosomes. One is more extra one chromosome. And sperm U, yeah, it has extra one chromosome. Explain the effect on the child is if the sperm U fertilized with a normal ovum. So let's take a look at the normal condition. Normal condition is N plus N, you become a 2N, which is diploid zygote, plus the sperm and the ovum. However, now is an abnormal situation. The sperm N plus 1 plus this ovum. So you get 2N plus 1. So you get one extra chromosome right there. So what is actually the effect? So in a normal situation, us human, for our somatic cell, we have 46 chromosomes, which is 2N equal to 46. So N, each of the gamete got 23. Each gamete got 23. But this one got 24 chromosomes. So 24 chromosomes plus a normal ovum that has 23 chromosomes, you know that you actually produce a 47, which is extra one chromosome. So effect on the child born if sperm you fertilized with a normal ovum. So basically the child will born with a genetic disease because it has an abnormal number of chromosomes. So the child will be born with genetic disease. So you can actually take an example of Down syndrome or Klinefelter syndrome. And because the child born, it has extra one chromosome. So it has a total of 47 chromosomes. So you can actually explain why this happened. So normally during spermatogenesis, something happens during the anaphase 1 or anaphase 2 where the chromosome didn't divide completely. Failure of sister chromatid to separate lah. So maybe one of them gets stick. That's why, yeah. So but this whole thing is actually called failure of sister chromatid to separate is actually called non-disjunction. Yeah, due to non-disjunction, or you can write it as failure of of sister chromatid to separate. Okay, this is quite K-back question, but it's actually a very hot topic lah. Okay, now let's continue for this. It shows two types of cell division. They want you to complete the table below to show the differences between them. So what are the differences between cell X and cell Y? So cell Y, you can basically see there's a homologous chromosome. Another thing to identify whether it's a meiosis, look at them. They have already crossed over and they have exchange of genetic material. So one of a small segment is white, another segment is black. So this one is definitely a meiosis. And then cell X is a mitosis. Why? Because during this metaphase, the chromosomes are aligned at the equatorial plane. But for metaphase 1, they are homologous chromosomes that are aligned. Yeah. So there are two types of cell division. One is mitosis, one is meiosis. So obviously, this one is mitosis and another one is meiosis. So the chromosome number in the daughter cell, how many will they lead up to? So for mitosis, it will eventually lead up to, it still stays the same, which is 6 of them. 1, 2, 3, 4, 5, 6. But for this meiosis, it will actually halve the number of chromosomes, become 3. Why? Because let's take a look at mitosis process first. 1, 2, 3, 4, 5, 6. Okay, anaphase, where they separated. So there'll be 1, 2, 3, 4, 5, 6 got put to the opposite pole. And then 1, 2, 3, 4, 5, 6 will be put to the opposite pole too. So in the end, it will form 1, 2, 3, 4, 5, 6. Yeah, it will actually form daughter cell with 6 chromosomes. And chromosome, you have to count based on the centromere. I know a lot of students get messed up. Okay, this one is actually 1 chromosome, 2 sister chromatid. And for this, it is actually also considered as 1 chromosome, but this only has 1 sister chromatid. But now we are actually focusing on number of chromosomes. The number of chromosomes still stays the same, just that the sister chromatid gets separated. So the sister chromatid right here, you can see 1. So you actually just count it based on the centromere right here. 1, 2, 3, 4, 5, 6. Yeah. So eventually it lead up to 6 chromosomes in each daughter cell. What about meiosis? So meiosis is a different thing already because for meiosis, the homologous chromosomes separate at first. So when the homologous chromosome gets separated, like that, they get separated. So 1, 2, 3. So this one also gets separated. So at meiosis 1, you can see that each cell already have half of the number of chromosomes. The number of chromosomes already being half. Yeah. So at first here got 6. Now the separation of the homologous chromosome lead to each of them having 3. And then it not only stopped until here, it still got the separation of so they will align for meiosis 2. Okay, for the anaphase 2 is where the separation of sister chromatid occur. But it still eventually lead up to 3 chromosomes right here. This one also same thing, they get separated. I know it's a bit messy, so sorry for that. So yeah, it eventually leads to four daughter cells, and each of the daughter cell still containing three chromosome, one, two, three, because we just count the chromosome by the centromere. So which is the moment where the number of chromosome get half? It's actually after the anaphase. The number of chromosome initially six, it already half to become three already. And the meiosis two, yeah, it still continue three. So meiosis one is a separation of homologous chromosome. Meiosis two is actually quite similar to mitosis, where it's a separation of sister chromatid. So yeah, that's actually lead up to this answer, like 6 and 3. And state the difference between chromosome behavior between cell X and cell Y. So this also mentioning about chromosomal behavior. So both are actually aligned at the equatorial plane. But the differences in between them is this one is chromosome, this one is homologous chromosome. So I will write it out in a table form, but you write it out in the complete sentence form lah. So basically, it is chromosome aligned at equatorial plane. Never mind, I won't be writing it in the table form. Wow. So it's at cell X. Wow. Homologous chromosome. We have to add up here. Homologous chromosome. Aligned at the equatorial plane. Or you can write a wave lah, at cell 1. And then for the spindle fiber, you can see that each chromatid, because here got two chromatid, each chromatid, okay, let me draw it out lah. This chromatid tied up, this chromatid also tied up. So it means that each chromatid is tied to spindle fiber. At the cell X, at cell X. Wow, what about this is actually each, the chromosome lah, each chromosome. One chromosome is tied up by one, and then this one also tied up by one. So chromosome at cell Y. And you can follow by another differences between them is the sister chromatid are joined together at centromere in cell X. While the sister chromatid of the homologous chromosome are joined together at the centromere. That's the small differences between them lah. But I would say that the most ideal answer is the first two lah. Because the third one is actually provided by the answer scheme. And while the sister chromatid of homologous chromosome. But I prefer you to refer it to the first 2 points to be better lah. Yeah. Ding ding. And then followed by this question, cell Y is exposed to radioactive rays. Yeah, they are hinting you that it's a mutagen and cause the component Z to fail to function. So when you're exposed to this radiation, yeah, it may hinder the process of it, lah, cause the something fail to function. So what is actually the component Z? Component Z is actually the spindle fiber that fail to form. So what happens next is the homologous chromosome fail to separate. Then yeah, it will actually lead to non-disjunction. So component Z is basically spindle fiber fail to form. So homologous chromosome, because this is about anaphase 1. So anaphase 1 is where the separation of homologous chromosome occur. Anaphase 2 is separation of sister chromatid. So homologous chromosome fails to separate. Non-disjunction occurs. And normally meiosis, it actually happens in the gamete cell. So you know that the gamete produced, it will be either extra one or less one chromosome. So abnormal gamete with less, it will lead to abnormal number of chromosome lah, basically. So are produced, are formed, or produced, and then yeah, it will cause a genetic disease. If abnormal gamete fused with a normal gamete, fertilizes with another normal gamete. So the one that you have the genetic disease is the offspring, means the child lah, of the offspring. In the offspring. Alright, it's basically chromosomal mutation lah, because it is a abnormal number of chromosomes. And by looking at this question, they show you information about this plant. It's among the medicinal plant that has been using treating side effect of menopause among humans. So menopause is when woman reaches around like 50 years old, there, around there, during aging lah, because there all the hormone is very low, especially estrogen, progesterone, then they will have all these menopause issues lah. Yeah. And realizing the importance of it, the plant is high demand, so the growth is very slow. Wire seeds and stem cutting at natural habitat and not sufficient to fulfill the need of customers. So they want to like boost the production of it. So based on cell division application, name one technique used to produce new plant. More of the plant to fulfill the need of customer. So this technique is actually quite widely used. Like, it's basically this tissue culture technique. So first of all, you state what is the technique first. Tissue culture technique. So what happens in the tissue culture technique? So first of all, to explain lah, they will basically obtain explant from the parent plant and they will go and cultivate it. Then they will place it in a culture medium and then in a petri dish lah, basically. Then it will keep the white wire mitosis to form a ball of cell, a mass cell like that, whereby it's called callus. And later on, the callus will differentiate into the specialized tissue of the plant. So it will eventually lead up to become a plantlet. So yeah, the baby plant just ta-da came out. So eggs plant are placed in sterile culture, or you can write petri dish. Sterile. Sterile means it's not contaminated. Okay. And growth is promoted by growth hormone. You can write auxin or cytokinin to be more specific. And this one, the egg plant will divide via mitosis to form callus, and the callus will eventually differentiate into plantlet. Divide via mitosis to form callus, and then the callus will differentiate to form a plantlet. But this one is way more detailed lah. The answer scheme actually just provided until here, and then they got right via mitosis. Yeah, so these three will do actually. Yeah, these four will do actually. Okay, and then moving on to this chromosomal behavior in every phase of animal, and this is obviously known is most probably a mitosis lah. So arrange the correct sequence of phases, because this one, yeah, they don't have any exchange of genetic material. You can't really see it well, and the way they divide, yeah, separation of sister chromatid, and you can see that their chromatid, yeah, they didn't have any exchange of genetic material, and they only have four phases. If it's a meiosis, they will actually try to emphasize the exchange of DNA segment or homologous chromosome or crossing over. So yeah, prophase, so prophase, metaphase where they are in the middle, anaphase where they are away, and then T. So it is starting from the first one and second one, third one, S. That is prophase, metaphase, anaphase, telophase. So explain chromosomal behavior at T. So it's basically um wait, yeah. So chromosomal behavior at T is basically the separation of sister chromatid. So sister chromatid separate and put to the opposite pole. Ta-da! That's all. Now let's move on to the next one right here. You can see that she has a malignant tumor around her uterus. So the cause of this condition is due to radiation. So there are actually types of radiation like X-ray, or you can write it as gamma ray, because they have a very high frequency. Means it can penetrate through anything like body cells. Yeah, it can penetrate through. Or from the sun, ultraviolet ray, UV ray. Okay. Now let's take a look. After 6 months, she had a bone metastasis. Means that the tumor will start to spread lah. It starts to spread already. It was found that malignant tumor cell from the uterus has spread to her bone. So initially it's at the uterus. Now it has spreaded to the bone, which is really serious. It's probably in a stage 3 or stage 4 already. So how it gets spreaded is actually through blood vessel passage. So how to explain in this manner by following this diagram, you can say that the malignant tumor, or you can mention the cancer cell, spreads through the help of blood vessel. That's how they get spreaded. Blood vessels and reach to the bone and lead to bone cancer. So the cancer cell, it will undergo uncontrolled cell division. To be specific, it is uncontrolled mitosis. It's not meiosis. Meiosis is for the formation of gamete and forms new tumor. So this is how tumor spread for a malignant tumor. Benign tumor is non-cancerous one. But this one is a cancerous one, that's why it's a malignant tumor. There are actually two types of tumor for your information. And as an oncologist, suggests to Mrs. K the appropriate treatment method to treat her illness. So during this type of situation, you can either use surgery. So surgery means you just cut away the tumor, or you can actually use chemotherapy or radiotherapy. So or maybe surgery. Yeah. Normally, if you do surgery first, then only chemo or radiotherapy to ensure all of them are being killed. Okay. Last but not least, for this paper, is actually a combination of Form 4 Chapter 6 and Form 5 Chapter 1. So it shows a longitudinal section of a root and shows a telophase stage at zone X. So zone X is actually the zone of cell division, right? And then followed by zone of cell elongation, then it only will start to zone of cell differentiation, it start to differentiate. So state the name of zone X, yeah, zone of cell division basically, right here. And then Y forms during cytokinesis. What is Y? So what happens right here is that the cell plate, right, it will, wait, sorry, the membrane enclosed vesicle will actually align at the equatorial and they will fuse together to form a cell plate. So Y is actually a cell plate. So what is the example of tissue present in zone X, which is the zone of cell division? So you try to think of a tissue that they kept dividing non-stop. I mean, they will keep dividing via mitosis. So that tissue is most probably a meristematic tissue. Do you still remember there is apical meristem, lateral meristem? Apical one is the one to grow taller. Lateral one is the one to grow thicker. Yeah. So it is the meristematic tissue. And then this farmer wants to produce a large number of quality breed durian trees in a short time. So explain this technique to the farmer. So it's basically a tissue culture technique. So this is very important for you to know this technique. So the first one, you just state what is the technique, tissue culture technique. Then the explant are placed in the culture medium. So the culture medium, of course, you need to provide nutrients, growth hormone, right? containing nutrient, or you can write it as, or you can write growth hormone. And then the X plant, what happens to it? The X plant will divide mitotically to form calyx. And then the calyx will eventually differentiate to form specialized tissue and become a plantlet. Okay, for this one, behavior of chromosome for cell A and cell B in two types of cell. So this is basically a homologous chromosome that are arranged in an equatorial plane. Cell A is a chromosome aligned at equatorial plane. So cell division, there are two types, mitosis, meiosis. So it's obviously know that this one is a mitosis and this one is the meiosis. And for the number of chromosomes in each daughter cell form, so mitosis, it will still maintain the number of chromosomes. So it is still, wait, yeah, one, two, oh, it is still four of them. The picture really look like three of them, man. It really can trick student, man. So one, two, three, four. Yeah. If you look further, right, it looks like three only. Okay. And for the meiosis, remember that number of chromosomes you get half in the end. So initially you got four chromosomes. You get half, become two. That's it. So that's all for this chapter. And then you have to put more focus in the mitosis, meiosis, and what happens for the tumor and the tissue culture technique, which is the application of mitosis. So yeah, that's all for this lesson, and then I will see you in the next video. Bye bye.